I'm at the Morton Arboretum, the lead institution for the Global Conservation Consortium for Oak, and also a board member of the International Oak Society. And if this is your first time, or if you're a returning webinar participant, welcome back, or welcome if it's your first time here. This is a webinar series that the iOS and GCCO co-host together, webinar number 10. And we, as always, like to just acknowledge the generous support of the International Oak Society institutional members, which you'll see on this slide here. So thank you all for your support. And thank you to our members too, of course, for your support as well and being a part of the network. And so just for the agenda for today, Roderick Cameron, also a board member of the International Oak Society. He will share some announcements to start, and then we'll go right into our presentations. We have the first presentation being Robinson Sudan, who's going to be giving a talk about oak diversification, specifically for oaks of Texas as well. And then Stephan, who will be talking about oaks of Lebanon. After each presentation, there will be time for Q&A. Feel free to put things in the chat too as we go along if you have any comments or questions, if you just want to put it out there. But of course, when we have our designated Q&A time, you can unmute and ask your questions or provide your comments then. But before then, please just stay muted while our presenters are talking. And then, yeah, I think that's everything. So, Roderick, I'll pass it off to you to share some announcements.
Sure. Thanks very much, Amy. Great to see everyone here. There's not much to announce. Some of us are recovering from the conference, which was actually last October, and do like to point out that I put on my conference T-shirt today. For those of you who are there or who have one, you'll recognize it. But we do have the Tour and Events Committee is busy planning next year, and we already have 3 events lined up. We're going to start off in the UK in May. We'll have visits to 2 oak collections there, to White House Farm, Garden and Arboretum, and Winkham Arboretum. That's May 16 and 17. And then in the Northern Hemisphere fall, we're looking at a tour of Turkey, in September, and then another one of Southeastern USA in late October, the second half of October, 15th to 22nd, and that'll be focusing on Georgia and Alabama, maybe also visiting sites in South Carolina and then northern Florida. You may have received, or you should have received an email about that just a few days ago, and we will soon be putting up more details on our website under the events section. So that's all I had. We'll go into our presentations. Remember, if you have any questions, general questions about the iOS, we can field those at the end of the presentation. So please do bring up any questions you might have about the iOS in general, about these tours and events. So back to you, Amy, and we can start with the presentations.
Great. Thanks, Roderick. Yeah, so we'll kick it off for our first presenter, Robinson. So whenever you're ready, Robinson, you can share your screen. Thank you. Great. Looks good. We see your slides. Yep.
Okay, great. Just the slides, right?
Yes, just the slides.
Okay, excellent. All right. So my name is Robinson Sudan, and I am a PhD candidate at Texas State University in San Marcos. And I am presenting today my dissertation research focusing on the mechanisms of oak diversification and some of the consequences of that diversification on Oak Gall Wasp communities. I got interested in oaks in part just because they're really interesting, but really because they're a great study system for some important evolutionary questions or processes. You can maybe consider the oaks here in the chapters as the characters rather than the setting for the story, because much of the real story is in how the outcomes of evolution have left their imprint on populations and how we go about observing and interpreting them. sorry. One of the real themes of that story is hybridization, which is becoming increasingly important for studying adaptation of populations and lineage diversification. Hybridization can homogenize and lead to local extinction of distinct taxa when hybrid fitness is greater than that of the parental species, the result being a hybrid population. We can understand why this could be concerning in terms of conservation and invasive species, but species can also benefit from hybridization when it involves a locally adapted species, as in the case of adaptive introgression, which is where adaptive alleles from one species are introduced into another species via hybrids reproducing with parental species. In this case, we have backcrossing of hybrids with species B and adaptive alleles from species A increasing fitness of species B. And this process can play an important role in species range expansion or in population persistence when environments or other selection pressures change. Beyond simply supporting the maintenance of biodiversity, hybridization can also result in the formation of entirely new taxa via hybrid speciation, when two interfertile species occupy distinct niches and their hybrids occupy niche space not exploited by either parent species. Hybrid speciation occurs when reproductive isolation develops between hybrids and both parentals. And here we're seeing hybridization plus niche differentiation yielding an entirely new taxon, and that's hybridization as an adaptive and diversifying process. In the case where hybrids exploit parts of the parental niche space, introgression can reduce fitness in parental populations and reinforce reproductive isolation between parental species. In this case, hybridization acts to maintain species boundaries rather than simply blurring them. Now, North American oaks are particularly well suited as a study system for understanding the role of hybridization plays in adaptation and diversification. They're speciose, ecologically diverse. Species within sections hybridize frequently in nature, and those interfertile species often occur in symmetry and or parapetry. And it's been hypothesized that one of the primary drivers of oak diversity here is hybridization and introgression, along with niche differentiation, which we've already seen can interact to produce hybrid species and reinforce reproductive isolation. And finally, North American oaks also provide an opportunity to examine how their own diversification can cascade to influence the diverse arthropod communities that they support. In the first two chapters, I directly address hybridization and adaptation in order to better understand the role of lineage diversification in oaks. I did this first by studying an interfertile 3-species red oak assemblage in the southwest US, and second by focusing on a much more speciose assemblage of southwestern white oaks. And in the final chapter, I shift gears a bit to explore the potential consequences of oak diversification across trophic levels in a southeastern US live oak gall wasp system. First, though, it helps to put these interfertile oak assemblages in a conceptual context centered on the syngamion. The term and concept were developed to describe a group of 3 or more interfertile species exhibiting complex and variable patterns of hybridization. And we know oaks are the exemplar of this phenomenon, and from my own observations of morphological intermediacy in southwestern red oaks, they certainly fit the definition, at least based on morphology. And those 3 red oak species in the Southwest are Hypolochoides, Gravesii, and Amorii. Their putative hybrids are largely distinguishable based upon variation in leaf shape, and conspicuously the variation in density of tomentum on the abaxial leaf surface, basically how fuzzy the underside of the leaf surfaces are, which is part of how we define hybrids in the field with those readily identifiable intermediacy of characters. So we have more than 2 species producing complex hybrid combinations. So we have, by that definition, a syngamion. And of course, this kind of thing is a headache for systematics and taxonomy, but honestly, that complexity is really kind of the whole point of the work that I'm doing. And so we might be able to look to the past to study the dynamics of syngamions, but evolution works the same today as it did in the past, and we can more precisely examine contemporary patterns of admixture in syngamions to understand their potential to generate novel genotypic variation, how introgression can vary when it occurs in a genome with 3 or more sources of ancestry, and how patterns of hybridization and introgression can vary with environment and species assemblage membership. So of those 3 known red oak species in the Southwest, Amorii and Hypolochoides are relatively widespread, and Gravesii is more restricted, found in far west Texas and the Sierra Madre Oriental of Mexico. They're quite morphologically distinct, as we saw with variation in leaf size, shape, and texture. Additionally, they show a good deal of ecological variation, with Amorii being more xeric adapted and Hypoleucoides gravesii typically restricted to higher elevations or sites that are more mesic or protected. Their areas of overlap also vary, and in the US, the only location where all three are found in symmetry is in the Davis Mountains, far west Texas. And that's particularly notable too, in that it occurs at the periphery of all three species ranges. And for the two widespread species, those are also quite disjunct populations. And for all those reasons, my research questions are focused on if admixture patterns vary between these peripheral and core populations, what that drivers of variation might be. So in the first chapter, I quantify patterns of population genomic variation and structure, determine levels of admixture among sympatric red oaks, and determine the role of environmental factors and geography in structuring population genomic variation and admixture. So the red oak sampling took place in two mountain ranges, the Davis Mountains of far west Texas, where they all three occur, and in the Chiricahua Mountains in southeast Arizona, where Amorii and Hypoleucoides co-occur. Collected a total of 768 individuals, identifying them to species in the field for morphology and assigning intermediate individuals as putative hybrids. In addition to leaf material for DNA extraction, I collected a voucher specimen for each, and GPS coordinates of individuals. Location data like canopy cover and photos of leaves and the plant as a whole were recorded using Esri field maps. I extracted DNA from silica dried leaves, produced a reduced representation genomic library, and after sequencing, reads were aligned to the Quercus rubra genome. Variant sites were called and filtered and yielded just over 21,000 genomic markers. In the population genomic analyses, I use entropy, which performs clustering similar to the program's structure to examine admixture proportions and interclass heterozygosity of individuals. And I'll use BGC, which is a Bayesian genomic Kleins model, to estimate rates and direction of introgression and to identify loci potentially experiencing adaptive introgression. So genotype environment association analyses, or GEA analyses, can identify loci associated with variation of environmental factors like precipitation or elevation, for example. In this way, they're useful in identifying parts of the genome where there might be signals of selection and associating them with their potential environmental drivers. I plan to use two complementary GEA methods, redundancy analysis, or RDA, and generalized dissimilarity modeling, GDM, and they're complementary because they model the genotype environment relations differently. So not all my results are in, but we can tell a lot from what I do have. We can start with this principal components analysis of the individual genotypes. So consider this as representing similarity of genotypes of the sample trees, which are represented as points on those plots. And this PCA shows quite a lot of hybridization among all three species and a lot of variability in degrees of admixture. So it sure looks like what you'd expect in a syngamion. And most of the hybridization is happening in the Davis Mountains, with evidence of three species ancestry indicated by individuals between the cluster of points forming that inner triangle. Also, our field identification clearly was not foolproof, even for these morphologically distinct taxa. We can get an even better picture of the population genomic structure of these assemblages by examining the admixture proportions from entropy. So each bar here represents a single individual, and the amount of each of the colors in the bars represents the proportion of the genome that a single lineage contributed to that individual. In this case, the three colors are for our three red oak species. They're arranged here first by field IDs listed along that x-axis, and within that by their collection location listed at the top, the Davis Mountains or the Chiricahua Mountains. And getting directly at one of my research objectives, these results confirm our morphological field IDs that the Chiricahua samples show a much lower frequency of hybrids. The two intermediate individuals all the way on the right side of the plot are in fact the only apparent hybrids. The rest of the Chiricahua samples show essentially no admixture. We can also see there's a very different pattern in the Davis Mountains, with many more hybrids and what are very clearly admixed individuals being misidentified by us as pure species. And confirming what we saw in the PCA plot, there are a number of hybrids showing ancestry from all three species there. And finally, what looked in that PCA a little bit like subtle but possible interspecific divergence between the two species that co-occur at both localities seems perhaps to suggest that it might in fact be due to introgression with Gravesii in the Davis Mountains, because almost all individuals of Amorii and Hypoleucoides there show some level of ancestry from Gravesii that's beyond, or appears beyond, that kind of background noise that you sometimes see with these clustering algorithms. So clearly there's active and relatively frequent hybridization in the Davis Mountains, which is very likely not the case in the Chiricahuas. And that raises a number of very interesting questions, namely what is driving the difference in frequency of hybrids? What are there environmental factors that are contributing to that difference? Maybe it's low effective population sizes in a peripheral part of all the species ranges. Is it adaptive introgression, or maybe even Gravesii acting as a conduit species, allowing consistent gene flow between Amorii and Hypoleucoides, but only in the Davis Mountains? Maybe it's some combination, as it often is. And how many hybrids are the results of hybrids mating with each other or backcrossing with parental species? Those patterns differ with three-way hybrids. And this sort of gets to the heart of how syngamions might differ in important ways from pairs of hybridizing species. So I'll address the role of environment with GEA analyses, as I said, and BGC will be able to identify parts of the genome which may be experiencing adaptive introgression, but BGC only works for hybridizing species pairs, and that clearly will not address all the hybrids in the Davis Mountains. I'll need to consider the best approach, though, for estimating effective population size and think more about Gravesii as a conduit species. I'll use interclass heterozygosity estimates from entropy to infer hybrid class for hybrids with two sources of ancestry. But again, the existing approaches also don't work for three species hybrids. But I do have a plan for that. I modified a simulation from Ben Wiens and colleagues to simulate hybrids, track their pedigree, and produce a genotype matrix that can be used in various analyses. So from real parental individuals from my real data, I created various hybrid combinations and conducted the same entropy analyses I used for the real individuals. So I have the same information, admixture proportion, and interclass heterozygosity for the simulated individuals. And the next step, which I'm working on now, is to use those data from the simulated individuals to train a machine learning classifier algorithm called Random Forest. After training, then I can ask Random Forest to assign my real individuals to hybrid classes based on the simulated hybrids. And conceptually, this should work because I know the exact pedigrees of each of the simulated individuals, and it should get to the core of how hybridization and introgression work differently in a syngamion. So that's where I'm now. And there's much more to do with this very intriguing system. So I hope I can share more results this when I have them. And then moving on to the second chapter, I'm essentially doing the same thing for the White Oaks using the same two mountain ranges, but there's at least one important difference that makes the White Oak system potentially more complex. And that's that White Oaks are much more speciose here than Red Oaks, and with quite divergent clades occurring in symmetry and parapetry. In the interest of time, and since I've just finished the sampling, I won't go into detail here except to share some numbers of individuals from each species by locality. And you can see there's some unevenness in the sampling, but part of that simply due to the low abundances of some of the species in these ranges, which is why I had to sample more individuals of those outside the target mountain ranges. So again, stay tuned for the results from this chapter, because I think they might even be more interesting and complex than the Red Oaks. Okay, so I'm shifting gears here a bit for the third chapter, where I focus on a group of wasps associated with oaks. But there is a clear connection here to the potential consequences of local adaptation and lineage divergence in oaks. We saw in the introduction how niche differentiation between ecologically divergent species can reinforce reproductive isolation between them. And this seems to be the case in two sympatric live oaks, Quercus virginiana and geminata. And these sister species are sympatric along part of the Gulf of Mexico coast, but they differ in their soil preferences. Virginiana also flowers earlier than Geminata with very little overlap. So they're both ecologically and phenologically divergent, meaning opportunities for hybridization are low, and any hybrids are less likely to be well adapted to the parental soil types. So this niche differentiation is important in that it facilitates co-occurrence of sympatric species, but you might ask why does this matter for gall wasps on oaks? And specifically for the gall wasps on these two species of live oaks, we need to look at the biology of those insects first. Gall wasps are highly diverse clade of phytophagous wasps that lay their eggs on plant meristematic tissue to hijack plant growth, forcing the production of abnormal tissue that acts as a shelter and food source for their larvae. They attack a wide range of plant organs from roots to stems, leaves, and acorns. They also are the foundation for a community of natural enemies that represents a number of trophic interactions among those guilds. These guilds include parasitoids of the gall-forming wasps, which utilize the gall wasp larvae as the food source for their own larvae, hyperparasitoids, which parasitize the parasitoid larvae, and inclines, which in this case use the galls produced by gall wasps as a natal chamber and food source for their larvae. So these galls are a part of a web of interactions which would not exist without them. On live oaks, some of the most widespread and abundant belong to the genus Belanocnema. And these wasps specifically lay eggs on very newly emerged live oak leaves, meaning a narrow window of available leaf resources each year. It so happens that in these live oaks, flowering time is highly correlated with leaf emergence in the spring. And this means that flowering time divergence in Virginiana and Geminata might directly impact opportunities for reproduction between Belanocnema that use each of these live oak species. And that's indeed what we see in those Belanocnema species that use these live oaks, both in a population genomic context as well as experimentally. And we see further evidence of this pattern among other genera of gall wasps on these same live oak species. What's even more fascinating, we also see this pattern reflected in inclines associated with these other gall formers, specifically a genus called Synergis. Some Synergis are also inclines of Belanocnema, but we haven't observed evidence for host-associated differentiation yet in those Synergis. So I just want to review here. We've got live oak species that differ in leaf emergence times, which is the critical resource for egg-laying opportunities in Belanocnema and other gall wasps. And these gall wasps show phenological divergence too, which depends upon the live oak species they're associated with. And Synergis, this common group of inclines that co-opt galls made by gall wasps, including Belanocnema, as their larval shelter and food, also show phenological difference with some gall wasp species, but the ones that attack Belanocnema have yet to be fully investigated. So that's a lot of interactions going on, and I just want to make sure that that is all clear. So in my third chapter, I asked, does host-associated differentiation in Belanocnema species cascade across trophic levels to drive divergent selection in associated Synergis species? To do this, I take a population genomics approach that will allow me to investigate the relative contributions and interactions of geography, host plant, and Belanocnema lineage on Synergis population genomic structure. Okay, so mature Belanocnema galls were collected from 35 sites across the range of 3 live oak species, reared at ambient temperatures, and Synergis were collected upon emergence, yielding a total of 768 individuals, and the same extraction and library prep protocols were used as in the first chapter. I used the same entropy and PCA approach as the first chapter, and just like the bar plot from the first chapter, each individual is represented here by a single bar, and the portion of the colors in each bar represents the proportion of ancestry that in that individual from the specified number of lineages listed in the vertical axes. And the best model, it turns out here, is 5 distinct lineages. To explicitly visualize that population genomic structure, I arranged individuals so they correspond to their majority ancestry. So this includes all individuals here arranged so they correspond to their lineages. We can easily see there's strong population genomic structure and very little evidence of admixture. The first 4 principal components from the PCA of genotypes explained over 84% of variance, and that is a lot. You can get another perspective from these PCA plots of the strong population genomic structure. So even though this and the bar plot give us important insights, we need to put these in another context in order to address the research question about the host-associated differentiation and geography in synergists. And so these are the same results from the first bar plot figure. I just rearranged individuals to visualize whether any of these groups are restricted to a single live oak species. That's part of what the annotation below the x-axis represents, but I also arranged individuals by the geographic location of their sampling site. So the dark to light from left to right in the annotation is west to east within each of the 3 live oak species ranges. And what does this tell us? It's pretty clear then that there's not a strong relationship between any of these groups and the live oak species they're reared from. In fact, 29 out of 35 of the sites had multiple synergist lineages, which means that for almost 83% of the sites, there are 2 or more sympatric synergist lineages utilizing galls of the same Belanocnema species. What's also clear is there's a strong biogeographic division centered around Ocean Springs and Gaucher, Mississippi along the Gulf of Mexico coast, which, with the exception of a couple of the fusiformis sites, is the only place where the Western groups overlap with the other Eastern groups. We can get a better look at this by visualizing the ancestry proportions by site on a map. So we can see well here that geography appears to be the factor most clearly explaining the population genomic structure of synergists. So at this point, we can say it seems unlikely that there's any host-associated differentiation at work in these synergists, but why might that be? First, they seem to have the capacity to utilize galls of multiple Belanocnema species, so are clearly less sensitive to the phenological differences in leaf emergence than the Belanocnema hosts. Second, it seems for sure like some niche partitioning is at work here too, given the multiple lineages found at a good majority of the sites and almost no admixture among them at all. And third, there may be some aspect of community ecology here that we didn't measure that contributes to the low host specificity. For example, Belanocnema leaf galls are known to be attacked by at least 28 species of parasitoids and equines, and annual insect populations are notoriously variable between years. So perhaps the apparent generalist strategy in synergists is in response to competition with other generalists or specialists, and it's some bet hedging strategy. But answering those questions are beyond the scope of this project. But what I will do next is conduct a GEA analysis to actually explicitly investigate the effects of geography and host plant in a modeling context. That will also let me explore whether genomic variation in Belanocnema shapes population genomic structure in synergists by using the Belanocnema genotypes from Amanda Driscoll and colleagues' research as a predictor in an RDA. So I started this talk highlighting oaks as a study system for understanding evolutionary processes, and I'm not alone in the view that they serve that purpose quite well. We saw in the introduction how hybridization can impact adaptation and promote diversification, which is thought to be a big part of the story of oaks. We've also seen how the process of oak diversification can cascade and drive further diversification in gall wasp communities associated with them, but apparently not in some synergists. But despite all this, we're far from any clear picture of how oaks became so diverse and what the full consequences of that diversification are for the communities they support. But what I hope is that this work will contribute to that knowledge and filling in parts of that picture we don't have yet. And not that anyone on this webinar here needed it, but I also hope I did some work convincing everyone that oaks are a fascinating setting for telling the story of how evolution works. With that, I'd like to thank Lilu, the best adventure dog in the world, and my always ready shadow, Kelsey Wogan, without whom I couldn't have done this work. The landowners, funders, and other folks that gave me motivation and inspiration, my family for being so supportive, and finally, the dissertation committee, particularly my advisor, Chris Nice, for his mentorship and showing me the ropes of population genomics. And with that, I can take some questions.
Thank you very much, Robinson. That was fascinating. I have a couple of questions that I'm trying to formulate, but if anyone else has others, please do speak up or put them in the chat. While people are thinking, what I had at the conference, I presented about Quercus maisie, and one of the things they found, which is a hybrid between two species that are no longer sympatric, Quercus gambili and Quercus macrocarpa. And one
Hmm.
of the theories is that, or one of the evidence that they once were sympatric and hybridized, is that there's a species of gall wasp that has evolved to be able to parasitize both. So does that mean that the gall wasps are like they can only hijack a certain genome, or a certain, I mean, or would they, or would it be limited to a section?
Well, typically it is limited to section. There is a lot of variation in gall wasp host fidelity. In some cases, it's dependent upon the phenology, like in the case of Belanocnema, of the meristematic tissue that they oviposit on. But honestly, it's really not entirely clear what the physiological hijacking process is. There's almost certainly a genomic component, but there also might be a chemical component, and that is what, as far as we understand now, that's probably the thing that's driving the section fidelity. Well, what's really interesting with the inquilines is that, this is speculation at this point for me, but I think an area of probably interesting study is that they don't face the same, because they're not making the galls, they don't face the same sort of physiological hijacking challenges. They don't have to stick to those adaptations. They just have to be able to find their host and be able to process the gall tissue and consume it without any obstacles, as opposed to whatever the process is during oviposition, they don't have to change the physiology of the plant. So there may be fewer hurdles to overcome in terms of host switching in that regard. Does that
Right.
answer it?
Well, yes, it does. But one of the things I've think about sometimes is like these oak collections that are being set up in places where the gall wasps don't exist, for example, in
Mm-hmm.
South America. If they ever were introduced, I think, which I'm not suggesting it should be, but if it ever happened, it'd be fascinating to see like what they would find to be able to live on, and if that would be, that might be field for further study.
I agree. I think that's, I've reared out a lot of, or I've grown a lot of West Texas oaks in Central Texas, and as of yet, over several years, I've not seen any galls on any of those West Texas species. So it may just be, it's almost certainly something we haven't identified yet that keeps local species from utilizing introduced species.
Sure. We do have a question in the chat. It's just come in from Liliana Duarte. How did you get to select these three oak species and Belanocnema kinsey?
Well, so there are three Belanocnema species that were recently disentangled using population genomic methods. And it turns out that there are, in fact, now three species in that kinsey, or actually the original species was treaty, Belanocnema treaty, that were split into these three different species, with kinsey and treaty associated with Virginiana and fusiformis, but there seems to be some break. Actually, no, I got that wrong. Sorry, Kinsey associated with fusiformis and Virginiana in the western part of the range, treaty associated with Virginiana in the eastern part, and I'm forgetting the name. And then a third species associated with Geminata. But these are the three live oak species in the Southeast US. I believe there's maybe some controversy over whether Quercus minima is a valid species or not. But was the question how I chose those? I'm
I
trying to
believe
open the chat
so,
now.
or why you chose them?
Um,
Uh, Hello,
Yes,
I
Liliana can,
yeah.
apologize.
yes, go ahead. Yeah.
Let me turn on my camera also. to be realistic, maybe better this way.
Great.
I was just questioning because I love all this world of galls and plants, and what I was asking, how did you start to do this selection? Not because there are so many galls in Quercus species. That's why this one. I am from Portugal. I don't know any of these Quercus. I don't know this Belaucnema. So that's why I was asking, just to know where to start if I want to do something similar in here.
Well, so I sort of picked up on the as a further step on research that had already been done with the Belknap-Nema, but I I know that they've served as a very good study system, given that they have fidelity, that at least this species complex has fidelity to the live oak section by Renties. So I believe that it just acted as a very specific, simplified is maybe the wrong word, but a very straightforward system with 3 host plants, and with the goal being to disentangle whether there was any of this host-associated differentiation, which I think was the focal point of the original research that one of my dissertation committees started. And then the opportunity came to study the inquilines because there's so many different parasitoids, hyperparasitoids, and inquilines that come out of these galls. And actually, the survival rate of any individual Belknap-Nema larvae in a gall is extremely low. So they ended up with all of these sort of ancillary specimens of the inquilines and parasitoids, and there's been just a ton of different work done on each of these. So asking this next question about whether the inquilines followed this pattern of host-associated differentiation was kind of the natural progression.
Great. Well, thank you very much. And thank you for your question, Liliana. We do have to move on to our next presentation. So thank you, Robinson. Wonderful presentation. And if you'd like to stop sharing your slides, we can
Yes,
get,
thank you.
thank you. We can get Jean to share his. And if you'd like to introduce yourself, Jean, please, when you start. You're still muted.
Hello, everyone. My name is Stephan. I am associate professor at the Lebanese University in Beirut, Lebanon, and I've been working on oaks since almost 10 years now. And I did present this first time on the GCO meeting in June, but actually it's the same. It's a presentation of the oaks of Lebanon. So in Lebanon, we have 7 oaks and, of course, hybrids. Eventually, we might have syngamions as you're suggesting. For this, I will move to the next slide. So the oak species that are recorded in Lebanon so far are Quercus caliprinus, which is a live oak, evergreen, and then Quercus infectoria, Quercus cherries, Quercus aethyborensis, Quercus luc, Quercus cedrorum, Quercus kochiana, and the hybrids. And those oaks are distinguished by a combination of traits related to fruits and leaves. Many are distinguished without microscopic analysis of the leaves, but it would be difficult without that. Unfortunately, Quercus libani, or the Lebanon oak, is not found in Lebanon. It's mentioned in the literature, but it's found more in Syria, Turkey, Iraq, and Iran. Now, what we have been doing in Lebanon, we started with the identification of some of the species that were forgotten by taxonomic revisions and other abandonment during the First War and the Civil War, late 20th century. So we went to the botanical description and identification of those species through various botanists. Minitsky, who did the oaks of Asia and Western Asia mainly, Paul Mutard, who did the identification of all the plants of Lebanon, Syria, and the neighboring countries. And then we worked with one of my students of the identification of our only endemic tree of Lebanon, which happens to be an oak, the Quercus kochianum. Then we did ecological niche characterization using Maxent for ecological niche modeling. So we visited all around the country to take occurrence or presence points of mature individuals of those oaks, because otherwise it would be difficult to identify them or discriminate them. And then using those presence points, we did ecological niche modeling using different factors, including climatic factors, topography, soil, and we developed the maps for their potential distribution. We also took samples from those trees during those visits, and we did a scanning for their morphological traits for the leaves first using digimizer, and we relied on previous publications to see what are the traits that we need to measure: depth of the sinus itself, the limb, length over width, etc., the petal and other parts of the leaves. But we didn't stop there. We took also fruits. So we had the acorns and the cupels, and we measured them in terms of diameter and depth. In parallel, we conducted molecular analysis using part of the plastome of the cells of the mitochondria, and we did it in collaboration with the colleagues from Italy, the team of Marco Simeoni in the University of Viterbo. And we compared the results of the molecular and the morphological analysis, and it happens that they were very well correlated, even for the hybrids. We started to do the predicted distribution for suitability under future climatic scenarios in collaboration with different colleagues. For white oaks, I'm working currently with Carlos Villavichosa from Porto, Portugal, and also with colleagues from Turkey. So I will start with the first species, which is the live oak. This is Quercus caliprinus. It holds one of those. The photo is one of the oldest trees in Lebanon. You can put almost 300 chairs beneath the crown of this tree in the shade. As you can see, it's a private property next to a house, but we still have big specimens like this one in the wild. But otherwise, it can be also reduced to shrub on shallow soils, dry slopes facing south, and areas subject to intense coppicing or forest fire. Many would confuse it with the Quercus coccifera, which is found in the Western Mediterranean basin, but they are totally different, and I hope we will be working on this on the long term to really segregate those species, not only from a morphological aspect, but also from a molecular approach. So as I said, trees can be as 15-meter height or more, but it's also very well praised for charcoal production, for fuel wood. It has a big cultural value because it is usually found next to churches, monasteries, or any other site of importance in the middle of a square in a village. It goes up from sea level to 1,500 meters in elevation in the mountain range, except in the northeastern part of the country, which is very arid, meaning that we have precipitations less than 300 millimeters a year. And then the honeydew from aphids provide a major source for the well-praised honey in Lebanon. And in a few cases, if the season is dry and there is no more fodder for the herds. Sometimes the shepherds might cut the branches to feed them to their goats, only goats, because sheep won't eat the leaves. And this will also keep them as low shrubs rather than trees in some parts of the country. Now, Quercus infectoria is different. Quercus infectoria is the next common tree in Lebanon, almost having the same distribution as the previous. It is also known as Quercus boissieri. It's a medium tree that can surpass 16 meters in height as well. It is a marcescent oak, meaning that it sheds the leaf just before the new ones appear in spring. So trees might hold their brown leaves all winter. We have also some monumental trees, and they can reach out 1,800 meters in elevation in their distribution, but they prefer deeper soils when compared to the previous species. Also found almost in all the country except the arid northeastern part, but also it's distributed in other parts of the Middle East and in Cyprus, Armenia, and Iran. Now, the Quercus cerris, also known as Iron oak or Turkish oak, and it is the tallest among the oaks in Lebanon, and it can reach 25 meters or more in height. A few monumental trees are preserved as those that you see in the picture in the snow, next to sacred sites or tombs or whatsoever. But the leaves are also used as fodder and the wood for fuel. It has a potential timber use because of this slender shape, and it's also attractive as an ornamental tree. So we find it only in a few sites with mesic conditions, enough humidity on the western slopes of Mount Lebanon, between 600 meters and 1,650 meters. We have the southern limit of the species, which span all the eastern part of the Mediterranean between Greece and Lebanon, but also in parts of the Balkans, Central Europe, in Austria, and in Italy. Quercus aethaburensis is another oak of the Cerris group. It is very specific to the Levantine countries, meaning Lebanon, Israel, Palestine, Jordan, and Syria. Eventually, it can be found in the Hatay region of Turkey. It has a very expansive, spready shape, with the branches going really wide rather than tall, as you can see in the photo on the lower part of the screen. And it is also a marcescent species with late winter leaf shedding. We have many monumental trees as well, and the leaves are sometimes triangular in shape. Sometimes they are not quite very easy to distinguish from other oaks, which also question the possibility of introgression or hybridization with other oaks like Quercus infectoria. It is mainly found on deep soils below 1,700 meters, mainly on the basaltic plateau of Akkar and a few sites in the south. The acorns, as you can see in the photo, are among the biggest between the oaks that are present in Lebanon. The cedar oak, or Quercus cedrorum, which is also a synonym of Quercus petrea, subspecies Pinnatiloba. It is a medium-sized deciduous tree, similar to Quercus infectoria, which we mentioned before. We have a few specimens that are still monumental with large trunks more than 1 meter. The leaves are very, very heterogeneous and variable in shape, and they can look something like Quercus cerris or something like infectoria, and sometimes in between. But they have symmetric regular lobes with more or less medium deep sinuses. The acorns are relatively smaller and conical, with a smooth cupule covering a third of their length, as you can see in the photo as well. They are very rare in Lebanon, and they are rarely used. Most of them are found in protected areas and next to the cedar trees, above 1,500 meters until the tree line, which is in Lebanon almost 2,000 meters above sea level. Outside Lebanon, they are found in Turkey and in Iran. So the species is substantially rare, and it's, I think, threatened at the IUCN Red List if we're talking about the subspecies level. Now, the look oak is an endemic species. It's endemic to Lebanon and Syria, to 2 or 3 mountain ranges, the Mount Lebanon, the anti-Lebanon, and the Druze Mountain in Syria, southern part next to Jordan. It is a small deciduous tree, sometimes shrubby with multiple stems, eventually due to past coppicing for fuel wood or to other stresses. It can reach 12 to 14 meters in height, with a few specimens that can have 1 meter diameter in terms of trunk. The leaves are lobed with irregular shape, and they are more acute at the lobes when compared to the ceres and to the Quercus cedrorum. The acorns are the widest among local oaks, with rough cupules covering almost half of the acorn. And as I said, they are endemic to 3 mountains in Lebanon and Syria. Quercus kochiana, or Kochi oak, it is attributed to Antoine Kochi, an Austrian botanist who contributed to the identification of the flora of the Levantine countries. And it is endemic to Lebanon. It is found in a few sites. It is endangered at the IUCN Red List. It was mistakenly named Quercus pinnatifida, but then it was rectified. Some trees can reach 20-meter height, and they can have a diameter of more than 1 meter. They are also rarely used as a fuel wood. Most of the populations are within protected areas. The leaves are deeply lobed, and they can even reach the main nerve of the leaf, as you can see in the photos. And they are strict to the western slope of Mount Lebanon, starting 1,600 meters up to the tree line, almost to the same area like Quercus cedrorum. In our study about the morphological traits, this is one of the scanned photos of those leaves of the various species. And we mention here 2 hybrids. And both hybrids, Quercus infectoria and Luke, or Quercus infectoria and ceres, could be eventually among the few hybrids between the white oaks and the ceres group. This was also confirmed by our molecular analysis later on. So those traits, sometimes they are similar to the parents, or one of the parents for, let's say, the acorn, and the leaves like the other parent. And sometimes they are like in between both. So in terms of morphology, we don't have really too much information how we can differentiate between these, but we know definitely that the parents, due to the presence of the parental species within the same niche or habitat of those hybrids. If we take the genetics in terms of analysis, you can see here the yellow is the live oak, which is quite diverse with different diversity between the different populations. It is followed by both Quercus ceres in the dark green and the white oak, which is Quercus infectoria. They are quite diverse as well. And you can see that the hybrids are those black dots that exist mainly between the white oaks or between the Ceres group in the middle in the green. Those groups can be identified. The Ilex group, which is represented by Quercus caliprinus in Lebanon. The Ceres group represented by Quercus ceres, Itaborensis, Luke, and the Lebanon oak from Syria, because we didn't find it in Lebanon. White oaks are represented by Amphictoria, Cedrorum, and Kochiana, and then we have the hybrids. In terms of their distribution based on the ecological niche modeling that we conducted, as you can see in the photo, we have some of them with very restricted range, like the Quercus cedrorum, the middle to the left, Quercus luke, and Quercus itiberensis on the top left as well. While the Quercus oak or Quercus caliprinus or Quercus amphictoria, Boissieri are the most widespread across the country. So taking this into consideration, we were able to conduct at national level their IUCN red listing, and also to define what are the priority species for conservation and for ecosystem restoration. Of course, those that are restricted are the most vulnerable to climate change and of highest priority for conservation. And thank you for your attention, and I'm ready for any question from your side.
Thank you, Jean. Great presentation. I'm fascinated by the oaks you have in your country, especially these ones with monumental size. They look just fabulous. Does anyone have any questions? Please put them in the chat, or by all means, unmute and ask a question. We have a question here on the chat from Robinson. Do any of these species have allopatric populations outside of the Mediterranean climatic region? And if so, do they show different patterns of deciduousness between populations? Jean, you're muted.
Sorry.
Yeah, there you go.
Well, outside the Mediterranean region, I'm not sure, and I've never actually searched for it, and I cannot tell if there will be different patterns of deciduousness between the populations. But within Lebanon, yes, we can observe for different species, different pattern of deciduousness based on the climatic conditions in some years, but also according to elevation. The issue is that Lebanon is a mountainous country with wide elevation, especially for species that can exhibit a wide range of distribution, like the Amphictoria. For instance, the Amphictoria, it may be deciduous on the highest elevation and marcescent at the lower elevations, where the leaves might stay on trees for a longer period. And also, according to annual variability in climate, if you take this year, we had an extreme drought event that didn't happen in Lebanon since the last 80 years, and I presume it's the same all over the Levantine countries and Turkey. And the oaks start turning yellow, like autumn or fall season, and as early as the beginning of August. So the deciduous pattern has changed due to drought rather than to cold conditions, which, as we know, is a pre-adaptation acquired capacity for all those deciduous oaks.
Thank you. So we can all see each other better, I think. Any other questions for Jean?
Yeah, here's one.
Yeah, Nicola, go ahead. Hi, good to see you.
Great to see you, Roderick. Thank you, Jean. It's a great presentation, really interesting. I really enjoyed it. You mentioned that Quercus ceres, or cherries, is associated with some sacred sites. I wonder if the trees are sometimes sacred, and if not, Can you explain a bit more about those sacred sites and perhaps recommend some literature or references about that, preferably in English?
Thank you, Nicola. Well, actually, sacred sites depend. You know, Lebanon has different religious communities. So whenever there is a land that belongs to, let's say, the church or a Muslim waqf, we call it, which a religious group land, it's not owned by the private people. It's rather owned by the religious people. So on those sites, trees are respected. So you will not see any goats coming or no one coming to cut the trees. And they end up by having monumental shapes because they've been there for the last 300-400 years. We didn't do any research or publication on that, but now with the IUCN launching this OECM aspect, and you have all those sites of cultural importance, which were exposed by colleagues from Italy in Cyprus last April, we will be interested to start making an inventory of trees. We started with smaller plants, especially endemics, in sites of cultural value, be it religious or other. Like Lebanon is a very old country. We have sites, archaeological sites from the Phoenicians, from the Roman time, and the Greeks. So basically what we are doing now within those archaeological sites where the government or the archaeological department is taking good responsibility of the sites, we are using those sites as ancillary sites to introduce and to protect critically endangered species. Now, for small plants, it's easy, but for trees, you know, they take more space. It would be difficult, but it is one option also to consider. And if I find any study that is related to the trees and those sites, I will let you know, because many of them are actually also in graveyards and they are well preserved. So religious includes also, of course, graveyards. Thank you.
And can you easily recognize those trees? Is there a way, or somebody has to tell you this is a sacred tree?
Actually, if the graveyards are very, very old, you can still barely see some stones around it. Near religious, church or monasteries, it's very evident. You have the building next to it. So it's very easily identified. You won't find them in the middle of the forest. They are always either near an archaeological site or near a human settlement or urban area.
Thank you, Jean.
Thank you for that question, Nicola. We have another question in the chat from Justin. Do you see examples in exceed non-hybrid species? Oh, you're muted, John. There you go.
No, Justin, all those hybrids that I have collected have lesser vigor than the parental species. At least those that I sampled and I verified. And with also minimal fruit bearing, sometimes absent, sometimes minimal.
Thank you. Another question in the chat from Harriet. Do you see any evidence of human-driven speciation or human-enabled distribution?
That's the big question. And thank you, Harriet, for this. I mean, definitely, this is the big question when you are in a place where cities are as old as 6,000 years. So human are present here with evidence of deforestation since the Holocene, meaning 8,000 AD. We started in the Bekaa Valley domestication of agriculture. And by studying in the sedimental soils pollinology and the concentration of pollens from species in Lebanon, Syria, Israel, and all around the neighboring countries, we found that between 8,000 and 6,000 years, we have a decrease in the cover of forest in agriculture area, and most of it was dominated by oaks at that time. So we're talking about 6,000, 8,000 years. There is a paper about this aspect in Iran, I guess, or all the Irano-Turanian region. You know, the Irano-Turanian region, I wish Parisa was with us today, but in all this Irano-Turanian region, it's dominated by steppe. I mean, the major biome is a steppe, which is a kind of similar to the prairies in the US, northern parts like Dakota, Nebraska, and those areas, which is kind of cold, dry biome. So Iran is a high plateau, also for Kurdistan and central Anatolia. And it's cold, but at the same time, it's hot in summer and dry. So basically, it's dominated by grasslands, which is the steppe. Now we have forests in some areas, and all of them are deciduous. None of them is evergreen. So it's not a Mediterranean climate. You will not find any live oak. It's not subtropical. But people start questioning whether those trees were planted or not by previous civilization for different purposes. Now we know that the Persians, the Assyrians came for Lebanon to take the cedars for their warfare, for their temples and constructions. But we have no idea about whether they have planted or tried to plant some of those species within their region. Now, definitely, there is an evidence of species distribution, assisted regeneration in some areas, but how much it has contributed to speciation, that will be really interesting to follow up. Thank you.
Great. We actually have Parisa on this call. I don't know, Parisa, if you want to add anything to that, if you're able to.
Hey, Parisa.
Yeah, I'm not sure. At least I saw it just now. Okay, well, wait. Any other questions for John? Okay, well, John, thank you very much. Very interesting presentation. It's always great to hear about oaks in different parts of the world, which personally I have not yet had an opportunity to travel to. Before we end, does anyone have questions about IIS in general or any other topic they want to raise? This is the opportunity to try to make this also like an IIS meeting, which okay, but if not, I'd just like to thank then our presenters, Robinson and Jean, for your efforts. These are really interesting presentations. Thank you very much. And thank everyone for thank you for attending to everyone else. And we will be having our next events next year. So with that, I'm not sure you're hearing. My connection is now unstable. I hope you all heard me there. Bye all.